get caught up, be caught up, and really ready to go. Beginning of the week, right? Pretend the exam is on Monday. Where would your mind be? Where would your kind of program be if the exam was on Monday? At that point, you just finish Monday, and then you have a couple more days, and then the exam. Okay? So stay on top of it, guys. Good luck. Good luck, and let us know. Let us know how we can help. Just a reminder that the exam viewing is continuing, and this is the last week. We'd like to finish that up this week. So please make time to visit either Oriana or Logan. Stop on by if you wanted to view your exam. Great way to kind of set the stage for the next exam. Just kind of seeing what you did right, what you did wrong, addressing questions about your approach to the exams as well as your studying. And yeah, we can talk about all of that stuff, guys. It's all on the table to talk about, right? So please, yeah, okay. You got the idea. Today we just continue and finish chapter 6, and then we'll start chapter 7. We conclude chapter 7 on Monday. All right, that's it. I had, I think, one student in my office hours on Tuesday. You got other things going on, I get it, but I'm kind of saying there's going to be very few supplemental lectures. I think this time, because I haven't had an audience in my office hours to do those kind of reviews, the supplemental lectures. So one more chance, I guess, or maybe two more chances as we approach the exam. I will be in my office tomorrow at 2 p.m. virtual on Teams. All right, stop on by. All right, that said, just some reminders. Of course, stay on top of your studying. Those reviews, complete them ASAP. Friday, the double helix quiz will drop and close by just a one-day quiz, a bonus opportunity if you've read the book. Or if you haven't, please, either way, complete the quiz. Sometimes I forget to post it. So if we're approaching noon and there's no quiz, let me know. I'm serious. If I forget to post it, my apologies, but just give me a gentle reminder. I'll be happy to drop that assignment. You have two things due on Saturday, the intervention number 3, right? And then, is it 3? Yeah. Yeah, thanks. And then mastering assignment number 4. Remember, that one's on chapter 6, which we're concluding today. But then, I've now opened mastering number 5 over chapter 7. Okay, mastering number 5 over chapter 7. I think, frankly, with the tutorials these assignments have, you can often complete them even before we completed the material. But if you'd like to wait till Monday, we're finishing chapter 7 on Monday. Okay? Just don't get them mixed up. Mastering 4 over chapter 6, mastering 5 over chapter 7. Okay? Monday. Do Monday. That's the last of the assignments. Okay? No more assignments prior to this next exam. Okay? No CDA, nothing. That's it. Okay? So you know the landscape. Go to it. Question about anything? Right. Who was the music by? Carlos Santana. All right. Just those of you interested. Okay. One of the best guitarists, I guess, according to popular opinion. Guys, we've been talking about biomolecules, right? In chapter 5, to be sure. But even in chapter 6, we're continuing this story of the biomolecules, right? And in case you haven't noticed, let's just revisit, right? So there's 4 main types of biomolecules. 3 of them are biopolymers, right? Proteins, nucleic acids, and carbs. All of them are rich in functional groups and are therefore hydrophilic and soluble molecules, right? And then we have this family of molecules that's hydrophobic, right? Basically everything in the cell that's hydrophobic we consider a lipid, right? And so that's the 4th and final category of molecule, right? You can recognize why I call them that, because clearly there's a discrepancy, a difference between those, right? So what we're talking about now actually is how these molecules are made and where. Of course, we said they're all built by dehydration. That common strategy of chemistry is used to build all of these. But where, right? Where in the cell cell does it happen? Well, let's think about this. So we've talked about the nucleus now, right, as a location. And indeed, there is much dehydration in the nucleus. And I think the two best examples, bless you, are the dehydration reactions that build what? DNA? Yeah. And RNA. Yeah, DNA is made from existing DNA. It's copied, and more DNA is made, right? DNA is made from DNA. RNA is made from DNA. Again, a copying of that DNA to make RNA. And both of those involve dehydration, as we will, we've talked about, but we'll also revisit before we're all done. Then we went on to the ER, right? Rough ER, smooth ER. The rough ER also does dehydration in order to make what? Rough ER? Protein, of course. It's covered with ribosomes. You should never forget what the rough ER does. It makes of course, it makes protein. Now, that's not to say protein doesn't get made in other places. In the first place, I think we should consider is the cytosol, right? Because there's ribosomes in the cytosol as well. But here's another location for protein synthesis: dehydration to make proteins on the rough ER. What does a smooth ER make? Lipid, right? We said this. Smooth ER makes lipid, and I just put biosynthesis because it can happen by dehydration, but maybe other chemistry as well. And we don't really talk about details here, but really all the types of lipids we talked about, the smooth ER is involved with that. Okay, so yeah, you get the idea. So we were talking in particular about the rough ER, and let's pick up there with the rough ER, right? So we had said that when it makes proteins, the proteins either remain within the ER. At that point, they can be ER proteins that do their job for the ER. We listed exactly one example of an ER protein. What was that? Oh, chaperones, I think, was the only example we listed. But the other possibility is that proteins made by the rough ER leave the ER. And how would you leave? In something called a transport vesicle. Right, remember, the transport vesicle will connect a number of different membranes or membrane enclosed structures or organelles, which we refer to as an indirect sort of connection by way of this transport vesicle, right? And what is it? It's just a compartment enclosed by a lipid bilayer, right? Enclosed by a membrane. And that compartment indeed carries protein around the cell. Okay? So either proteins stay in the ER or they leave by transport vesicles, right? Within transport vesicles. Okay? And the last thing I think we said was that there's a primary route of traffic from the ER to the Golgi apparatus, as it's called, another membrane enclosed compartment. And I guess that begs the question then, what is that and why does that happen? But any questions about that? Because that's where we left off on Monday. Okay, no? Let's talk about it then. So this is the only organelle of which I'm familiar that's actually named for a person, right? In this case, Camille Golgi, Italian psychologist, I guess observed it and characterized it, and what a striking structure it has. And this was the very end of the 19th century. What is it? Well, it is indeed a collection of sacs, right? Which, again, we mean a membrane enclosed compartment, right? A collection of sacs which are long and flat, right? So multiple flattened sacs would be one description of the Golgi apparatus. Now, through many years of study, again, we found that these sacs contain enzymes. The sacs contain enzymes, and they're different between the different sacs. So different sets, or a unique set of enzymes within each set. And that begs the question, what does it do? What's the purpose of this Golgi? Well, we already said it will receive transport vesicles from where? From the ER, right? What we found is that it can also transmit transport vesicles from sac to sac. And so the sacs can effectively talk to one another by transport vesicles. And then it will bud off vesicles to yet other destinations. Okay? So receive, transmit, and then bud off vesicles. So I think I said on Monday, all of these diagrams, or rather micrographs, incredible. I mean, just beautiful to look at, but then the diagram that accompanies will probably be a little easier in terms of really understanding what it is. All right, so here you can see more clearly, I think, that it is multiple flattened sacs, and that vesicles are fusing with the Golgi on this side, right, and then can be transmitted, right, can actually go from sac to sac. You kind of see that here, I guess maybe here, right, and then leave on the other side. So note the red arrow. So we have a flow of ER products, proteins, through the Golgi by this vesicular traffic. Okay? Is that clear? I like this one in terms of the clarity. Yeah, but what's the point, I think, is the question. Why? I mean, so what are we saying? ER proteins are going to come here and then leave, right? So what's the point? Why even have this step or this stop along the way if you're going to go someplace else anyway, right? Well, no, this is there's a purpose behind this. What we found is that the enzymes within the sacs will work on ER products, modifying them. So the enzymes will modify ER products. Okay, so we can call that processing. The Golgi processes ER products, which, I mean, biochemically we mean that to mean chemical reactions, right? It will do chemistry on ER products, again, with those enzymes. It will then sort ER products. Okay, this is not obvious from the figure now, right? So you get all sorts of ER products, all kinds of different proteins coming in, but when they leave, they're sorted so that perhaps one vesicle has ER product A, protein A, which it's loaded full of, and then another vesicle may be protein B, right? Another vesicle protein C, and another vesicle maybe proteins D, E, and F, right? But sorted by the Golgi. That's the job, okay? So to process, sort, and then ship those ER products to another destination, right? A destination which needs those proteins, which expects those proteins in order to do its job. Oh, so important component of the endomembrane system. Question about the Golgi. All right. Well, then the question, I think, should be, where do vesicles leaving the Golgi go? What are the possibilities? Well, we would like to consider two routes of traffic from the Golgi to other places. And these are, one, to the cell membrane. Vesicles leaving the Golgi can fuse with the cell membrane, at which point the contents of the vesicle can what?
Get secreted.
Can actually leave the cell, right? So the cell membrane, is a point of entry and exit from the cell. So if you take vesicles carrying proteins to the membrane, then the proteins can leave the cell. Okay? So this is known as secretion. When cells produce a product and then export it in this way, that's called secretion. Secretion. So one route of traffic is really to cell membrane for secretion. Secretion. A secretary route, if you will. And the other route is to another structure we're yet to talk about called the lysosome or lysosomes. They could invent lysosomes to another organelle called lysosomes. All right, let's get a figure in here now to kind of demonstrate this vesicular trafficking, right? Again, from the ER, beginning with the ER, right? Vesicles to the Golgi. Oh, by the way, you can kind of reference the two sides of the Golgi based on their proximity to the ER, right? So the face of the Golgi that's facing the ER is called the what? Cis. Yeah, cis face. Whereas the opposite face, the opposite, the one that's across the Golgi from the ER is the, of course, trans face, right? Again, across from the across the Golgi from the ER. Okay, so that's a nice sort of a terminology kind of reference where you're coming or going from. Again, vesicles can leave the trans Golgi and arrive at the cell membrane, right? Number one. Where what? The products. Yeah, this is really, folks, I got this figure from another text can you see what's happening to the contents of the vesicle here? Right, so when the vesicle fuses with membrane, whatever is in the vesicle ends up outside. So what happens to a vesicle's contents? I don't know if you can see that, so I added one more. It ends up outside the cell. Okay, there's a protein that ends up outside the cell. All right, so we have just explained how our body's proteins get to be outside of cells, if you are a protein outside of cells. So for example, a digestive enzyme in your mouth or stomach or small intestine, that has been produced by glandular cells and exported from those cells through this mechanism we call secretion. Okay? Digestive enzymes. Protein hormones, which are in your bloodstream. They're not in cells. They are made, again, by glands and then secreted into the bloodstream. But again, that involves a manufacturing within cells of the glands and then an export into the bloodstream. Okay? Antibodies, you have some familiarity with from quiz number 4. Made by a type of immune cell. Again, at the rough ER, which then gets processed in the Golgi, shipped to the membrane of those cells, and then ends up in the bloodstream. Okay, so we just explained a profound answer, I guess, or a profound answer to a profound question, which is how our body makes proteins and puts them where they need to be, outside of cells. Okay, proteins outside of cells. Does that make sense? Okay. All right. Number 2. Destination, again, you don't have to leave the cell, and instead you could go from the Golgi to the lysosome, which I guess we're yet to talk about. And unless there's a question, let's talk about lysosomes. What is that organelle? So, remember, lyse means to break, right? Remember, hydrolysis is hydro and lysis, breaking with water. So this is a body, a some, or a body that's capable of lysis or hydrolysis. Okay, so the name actually tells you what it does. So it is a membrane enclosed sac, which ultimately almost everything is a membrane enclosed sac, which contains enzymes that do hydrolysis, hydrolytic enzymes that do hydrolysis. Okay, so again, like the Golgi, they contain enzymes, but these enzymes, not merely processing them, hydrolysis, they're breaking down their targets. Moreover, the lysosome is capable of fusing with compartments which originate from the cell membrane. Okay, so lysosomes, which have enzymes in them, can fuse with vesicles or larger structures we call vacuoles. So these terms kind of specify size, a vesicle or a vacuole, even larger structure originating from the cell membrane. What's that about? Well, let's talk about what a vacuole is first and then see if we can connect the dots here. Okay? So this term vacuole, again, it sort of implies a size larger than vesicle. Again, a membrane-bound sac with some specified function in the cell, depending on the vacuole. I think you're familiar already with a vacuole in plant cells, which is a very large one. Okay? Vacuoles are large, but I mean very large. The plant central vacuole is a large vacuole. And what does it do? Increases
surface area
the surface area to volume ratio. Okay, I said that this was a topic you're responsible from the lab. Okay, so I'll kind of leave it there, but I'm telling you that this structure, by taking up space in a plant cell, is effectively increasing the ratio of surface area to volume. Right, it's decreasing the cytosolic volume, therefore increasing that ratio. Okay, so that's one type of vacuole, but another type, more relevant to our present conversation, is food vacuole. This is a vacuole that buds in from the cell membrane and then fuses with lysosomes. Okay, so that was the connection, right? Buds in from the cell membrane, then fuses with lysosomes, okay? Can anyone think of a cell type you know of that can bud in a vacuole like we're describing? Yeah. So neurons, not to my knowledge, which doesn't mean it's not true, but maybe something you've seen in lab, found in pond scum, maybe? Amoeba, yeah, nice, very good. Amoeba can swallow another cell, I think, right? Or smaller particle. That's what we're talking about, creating a food vacuole by swallowing. Yeah, amoeba does that. It makes a food vacuole. Yeah.
Isn't that a type of phagocytosis?
It is called phagocytosis. That's right. It is phagocytosis. And I put a ellipsis here because we don't have to go to pond scum to find this functionality, because you have cells in your own body that do this. You have cells of the immune system that are capable of swallowing bacteria, right? What is that called? Macrophage, right? The literally big eaters, right? Macrophage, big eaters. Well, aptly named, considering they swallow other cells. Yeah. Okay. So what we're saying then is that these cells that create food vacuoles, then the food vacuoles become fused with lysosomes full of hydrolytic enzymes. So the function of the lysosome then is clear. It breaks down the contents of the food vacuole, breaks it down by hydrolysis. Is this clear? Yeah, so how do you use food? You break it down, right? You have to hydrolyze it, and cells can do that. Some cells can do that, anyway. Another function of lysosomes is to break down the cell's own parts. Yeah, this is another important function, to break down the cell's own parts, to break down organelles. Effectively recycling them. Okay, this is not deleterious to the cell now. This is not harming the cell. It is recycling, right? So if an organelle becomes old or damaged, unproductive, you could break it down with a lysosome and then release the monomers. Break it into monomers, release the monomers again, and then recycle that raw material. Okay? This is called autophagy, which is eating yourself. Right? The cell is eating parts of itself. Autophagy. Yeah, really big function of lysosomes. All right. Question? All right. So I have this impressive image of a white blood cell, right? A macrophage. Clearly a macrophage. You can see in the dark stain here the lysosomes, right? So this thing is full of lysosomes, as it must be if it's going to engulf cells and then break them down. Okay? And then this is a micrograph of autophagy, right? So the lysosome here has swallowed not one but two organelles, one of which you may be familiar with, these, the mitochondria, right? And now it's going to break them down. Again, recycling their components into raw material. Okay. Cool. All right, so one diagram here just kind of show the whole thing. So again, we said that vesicles would leave the Golgi and then fuse with lysosomes. Lysosomes in turn fuse with vesicles or vacuoles originating from the cell membrane, right? We call those food vacuoles. And here the two fusing together there, and then the enzymes go to town on the food, right? And again, products of digestion, breaking it up into monomers. Okay? Question? All right. I have a question. Oh, okay, so what was the deal then? Why is the Golgi sending ER products to the lysosome? What is that about? What's the simplest answer based on what you've learned?
So if you recycle them and then break them down.
So yeah, you could go that way, right? So you could say maybe there's a reason to recycle them, but I mean, we just made it, right? It's just the ER just made that protein. It went to the Golgi and then went to the lysosome. So what? So tell me this: where do what are the lysosomal hydrolytic enzymes? What are those? A type of proteins. Right. These little dots, the enzymes, are proteins. How did they get there? They were made by the rough ER, sent to the Golgi, processed, sorted, and shipped to the lysosome. We just explained where lysosomes get their enzymes. And now we're saying it, so it's clear. Okay? All right, so why send proteins there? That is the source of lysosomal enzymes, the source of the lysosome, essentially. Okay? Another question, why don't these enzymes, which are so good at hydrolysis, and by the way, they do it like every kind of biomolecule they can break up. They can break up protein, they can hydrolyze lipid, they can hydrolyze carbohydrate. They just great enzymes of a diverse variety. Why don't they damage the ER or the Golgi before they get to the lysosome? It is the environment, yeah. So the enzymes of the lysosome are not active until they get to the lysosome. They're not active in the ER. They're not active in the Golgi, where there's all those other proteins that they can damage, or even the ER Golgi itself you can damage. They're not active. They're only active when they get to the lysosome, when they need to be active. And that control is indeed established by the environment of the lysosome. Okay, so what about the environment? pH. pH, right? What is the pH of the cell again?
7.
It is 7, but the lysosome, 5. Yeah, pH of 5. So it's 100-fold higher proton concentration, at which the enzymes charges change a little bit, and they arrive at an active conformation. The enzymes snap into an active conformation. Okay? So it's a control by pH, which is something you come to understand is important for protein structure. Last but not least, what does all of this functioning of lysosomes have to do with digestion in your GI tract? Is there a connection? No. No. No, this digestion, folks, is in your GI tract. This digestion is intracellular. Okay? And again, we've described how it's important in certain cells, particularly in certain cells. Okay, so this is a very specific type of intracellular digestion, not to be confused with this, okay, digestion in your GI tract. This digestion requires that enzymes be made, but then secreted from the cell into the GI tract. Okay? So this is not digestion that you think of like this. Okay? Is that clear? All right. So I think the point is that I guess we as multicellular animals can do digestion in a space within us called the GI tract, but also can do it within some cells like white blood cells, like macrophage. Okay, very good. Lysosomes are a type of organelle that are more frequent in cells that use them. We just said macrophages are rich in lysosomes. I think some cells don't even have lysosomes, and so please be aware of that variation in the case of lysosomes. Okay, question? Folks, that then brings us to the end of the endomembrane system. And at this point, we can recognize the importance of this system in the production and processing of proteins, right? It's going to manufacture using its ER capability. It's going to sort ER products in the Golgi and then ship them to other destinations. Even vesicles are part of the endomembrane system, right? That destination might involve leaving the cell, right? So this is a way to get things out of the cell, but it's also a way to get things in the cell, right? Because whether the vesicles arrive at the cell membrane or formed at the cell membrane will dictate whether something is leaving or entering. So we're talking both export and import. And then in the way of the lysosome, we talked about a recycling capability. Folks, this is the manufacturing component of the eukaryotic cell. Okay, what is the EMS? It's the factory of the eukaryotic cell, the factory component. It does all that. Question. All right, so you see why I said this is a topic that is not only interesting, it's complex, and it's a lot to consider. All right, and if there isn't a question, think about it, let me know. We did have a prelude, I think, to this section. Let's do an epilogue. Bound ribosomes and free ribosomes are the two types of ribosomes we said. So let's be clear now. Bound ribosomes make proteins for whom?
EMS.
The endomembrane system, right? They're making proteins for the ER, like those chaperones. They're making proteins for the Golgi, like those processing enzymes. They're making proteins for the lysosome, those hydrolytic enzymes. So we've explained how all of these organelles get their proteins now. They also make proteins for the cell membrane, because if you arrive at the cell membrane in a transport vesicle, but you don't leave the cell, you do have the ability to remain just part of the cell membrane. So this is a way to get proteins into the cell membrane. So we've explained that as well. We've also explained proteins leaving the cell, like digestive enzymes, or protein hormones, or antibodies, any number of other intercellular proteins. Oh, bless you. And then there's free ribosomes. What do free ribosomes make proteins for if all of that is done by bound ribosomes? The cytosol. Nothing important, just that rich, complex biochemistry of the cytosol. Yeah, the cytosol needs proteins. It's full of proteins and enzymes. It's also going to make proteins for any organelles that are not part of the endomembrane system, right? So things like the mitochondria, which we haven't talked about yet, the chloroplast, which we haven't talked about yet, peroxisomes, which we will not talk about, but none of these are parts of the endomembrane system. They need an alternative source of their proteins. Okay? Question? All right, well, why don't we ask this question then? What energy drives cellular work?
ATP.
ATP, of course. And that's a segue then to our next bullet, which is mitochondria and chloroplast. All right, so these are organelles we're going to talk about more, and like a lot more, down the road. So let's just basic structural features and then move on, all right? So both of these types of organelles are involved in converting energy. Right? And I added here the first law because I think in the science of energy, which is called thermodynamics, the first law is that you can't make energy. You can only convert it from something that's already there. You can convert energy, you can't make it. So that's what these organelles do. They're energy converting structures. And as we just said, they're not part of the endomembrane system. And so they're not part of this vesicular traffic, right? They don't receive vesicles. They don't form vesicles. Moreover, they are semi-autonomous in that they manage themselves, actually. They have their own DNA. They have their own, say that again, they have their own DNA, and they have ribosomes in them, which suggests what? They can make what? They can make protein, yeah. They can make some of their own protein, not all, but some of their own protein, they can make it, they do make it, okay? So again, a semi-autonomous, kind of separate from the cell itself quality. Okay, let's talk about each now in particular, just a little bit, starting with mitochondria. Mitochondria are going to convert sugar energy into ATP energy. So the one that literally makes ATP is mitochondria, right? Mitochondria make ATP. And then here is a stunning electron micrograph of a mitochondrion, right? How many membrane enclose the structure?
2.
It is 2. Okay, and here you can see this, right? So there is a outer membrane that's smooth, and then an inner membrane that is like highly folded, right? All of these folds are part of this inner membrane. Okay, so 2 membrane, outer and inner, smooth and folded respectively. Mitochondria can vary in size, usually from 2 to 8 micrometers, I believe, and their abundance varies depending on the energy needs of the cell. Okay, so we already said that the particular type of cell we have are really full of mitochondria. What was that? Muscle cells, yeah. Liver is another good example. I think a liver cell can have 1,000 mitochondria. One liver cell, I think
That's a lot.
1,000 or more mitochondria. And so it just depends on your energy needs. Okay? Mitochondria are found in animal cells, but not plant cells. True or false?
False.
That's not right. It's a question. It's not right. Plant cells have mitochondria, and suggesting they don't is like saying plant cells don't need ATP. Of course they need ATP, and they make it with their mitochondria. Yeah. So just don't develop an either or, like you have to have mitochondria or chloroplast. You can't have both because plants have both. Okay. All right. Good. False was the answer. Okay. So going on to chloroplast. Again, an energy conversion, this time from the energy of light. They convert the energy of light into the energy of sugar. So, speaking of preludes or prequels, right? This is before the mitochondria. Before the mitochondria even gets that sugar to make ATP, you need a chloroplast to make that sugar, right? To make glucose. All right, so a little bit of terminology here, which we don't need to get into at the moment, but yeah, how many membrane enclose this organelle? It's harder to see, in fairness. It's 2 again, all right? It's 2, but they're like right on top of each other. So there's an outer membrane and an inner membrane, but they're like, side by side. They're right on top of each other, so you can't really distinguish them. I can see them from where I am, but I think you may not be able to. Interestingly, there's a 3rd membrane enclosed compartment floating within the chloroplast. Okay, it's not enclosing it, so I'm going to still say it's 2 membranes enclosing it, but this 3rd membrane is sort of within it like an organelle, like an ER. It's really like an ER in a chloroplast, and that's actually a key component to. So you know what this process is, right? This is photosynthesis. Of course, chloroplasts do photosynthesis, and this 3rd membrane is like the first part of, it's the light harvesting part. It harvests the light, does the first part of photosynthesis. Okay, they're a little bit bigger now than mitochondria. I think on the average 5 to 10 micrometers, and not all plant cells have chloroplasts. What plant cells have chloroplasts? Oh, mesophyll, is that what you said? Yes, they're called mesophyll cells. They're found where in a plant? In the leaves, right. You're so fancy. Right, they're mesophyll cells, but they're part of the leaf. So they're leaves. They're found in leaf cells. Okay? Not all the parts of the plant, and certainly not the roots. What will be the point? How do you harvest light if you're in the roots? You can't, right? So this is an organelle, again, that's found only where it's really needed in plant cells. Okay, question? All right, so as I said, to be continued, because we'll talk more about mitochondria and chloroplasts down the road. But I did want to say one more thing about them before we leave them altogether. What is the origin of these organelles? Right, so there's a model that describes where they originated. And the short of it really is that they were once ancient bacteria, right? So not recent bacteria, way long ago, right? They were ancient bacteria, and they were aerobic, right? They were able to use oxygen to make ATP, which is what your mitochondria does. Okay, and then we got that capability actually from these bacteria essentially by integrating them. Okay, so I know you've studied this in the lab as well, but the endosymbiont theory suggests that an ancient eukaryotic cell engulfed an ancient bacteria that could, for example, it could make ATP, right? And then suddenly, not suddenly, but maybe eventually got the ability to make ATP itself. Okay, so, well, that's an interesting idea, to be sure. What's the deal? Well, I mean, the deal is it's a mutual benefit, right? So the larger cell gains the ability to make ATP, or maybe make more ATP than it was previously making. The smaller cell is protected from the environment within the bigger cell, so that could be an advantage there. Moreover, there's all sorts of supporting observations for this, and I just listed some of them here. The shape and the size of a mitochondrion is very similar to some bacterial cells, like a rod-shaped bacterium. The model of endosymbiosis can explain why you get multiple membrane instead of just a single membrane, right, in this way. We already talked about how these organelles, both mitochondria and chloroplasts, are semi-autonomous. They have their own DNA, like cells. They have the ability to make protein. They have ribosomes. And those ribosomes are different from the cell's ribosomes, actually. They're more like bacterial ribosomes. And then modern day examples. So, you might ask, yeah, okay, all that's great, but, do you ever have does one cell ever swallow another cell? Well, we just talked about how our own white blood cells can swallow another cell. So there you go. We can do that ourselves. Moreover, there are examples in the modern day world where one cell will swallow another cell but not break it down, and they'll kind of live together in that way. So there are modern day examples, not to say that it becomes a mitochondria, but the idea was that it happened, in ancient history, and then over time became something else. Okay, a lot more to say about that, but that's just what we think. Okay, question? Okay, if not, then I have a question. What are these? Different cells, right? Okay, so we don't even have to look at all of them, but herbaceous neuron, muscle cell, and some blood cell there, plant cell. They're different cells, and the point I want to make here is that cells have some really different structures, right? I mean, every textbook presents a cell like it's a round, kind of amorphous kind of thing, but real cells have all sorts of different shapes, which then begs the question, how is that even possible, right? What is it that gives cells all these different shapes, right? And the answer, they have a skeleton, right? Cells have a skeleton, just like we have a skeleton that gives us our overall shape. So plants have a skeleton. We call it the cytoskeleton. There's a little more in the posted slide that I care for you to know, honestly. So just be aware, the cytoskeleton is a network of fibers spread throughout the cytosol. It gives the cell shape and support. What's it made of? Protein, yeah. Just like our skeleton is made of protein. in addition to calcium, right? It contains protein. So are cell skeletons made of protein, just protein. Okay, they're protein skeletons. Moreover, the cell skeleton can do a lot of other stuff for the cell. Okay, just like the cell membrane is not just about a barrier, right? The cytoskeleton is not just about that. You can transport stuff within the cell using this network like like railroad tracks, right? Transport what? Well, protein, whatever's in vesicles. How do you think vesicles go from point A to point B? I guess it's diffusion, right? No, vesicles move along cytoskeletal tracks, which guide a vesicle to where it needs to go. So transport within the cell requires a cytoskeleton. And movement of a cell, movement like the flagellum, you think of that? These are all offshoots of the cell skeleton itself. So these kind of movements of a cell come from the functioning of the cell skeleton. Right, so it's very dynamic. It's a thing, it can move, it can change shape, it can even restructure into something else entirely.
Mm-hmm.
Okay, I'd like you to know this part. A cell skeleton can restructure into the components needed for cell division. Okay? I'll give you an example. I put this figure in here because it kind of shows how the skeleton gives shape and support, but then there's some components that are sort of within the cell, kind of spanning back and forth there. So there's three different protein fibers that make up the cell skeleton. We don't have to get into the detail here, but please know microfilaments are important for animal cell division. And that's because they restructure into a ring that contracts. Okay, microfilaments are part of the cell skeleton that restructures into a ring that contracts. Microfilaments are great for contraction. Any kind of contracting, like your muscle, any kind of contracting movement, you can pretty much assume is involving the microfilaments. In this case, it's a ring that contracts and pulls the cell membrane along with it, and that's how the cell pinches in two, is that action of actin filaments. Okay? So know about the contracting ring, and also know microtubules, which are thicker than microfilaments. Microtubules are important for cell division because they make up the fibers of the mitotic spindle. Right, you've seen that before, right? The spindle fibers that pull apart a chromosome, what are they? They're microtubules. Again, a repurposing of the cell skeleton, or the microtubule part of the cell skeleton. Okay? Question? Okay, so that's what I'd like you to know, especially about the cytoskeleton. There's a great animation from Pearson that shows all the stuff we just talked about in this chapter. It's under the exam. It's in the module. It's in the chapter 6 module. So take a look. It's really cool animation. And why don't we turn to? Turn the page. It's a new chapter. Question? All right, let's get a start on chapter 7, and we can pick up and conclude on Monday. So again, cell membrane, nothing but in this chapter. We will revisit the structure of the membrane and then talk about what makes it permeable. Remember, this is not just a barrier, we're able to import the nutrients that the cell needs. There's actually a couple of ways to move solutes across the membrane. One involves existing energy, if I can say it that way, involves existing energy, and then one requires an input of energy to happen. So these are passive and active transport mechanisms, and we'll talk about that. And then at the very end, we can revisit vesicle-mediated transport, but we don't need to almost because we just talked about it. You already know you can move things out of the cell or into the cell using vesicles, because you've seen that. You've seen examples. Okay. All right, so we got epithelial cells, right, from the, I guess the lining of the cheek, epithelial cells. I put this here, and everyone has seen this at some point, probably under the microscope, because it's not rocket science to recognize there is a barrier, right? There is a membrane enclosing the cell. Clearly, something is there, but what is it? That's another question. This was actually a compelling question in cell biology. I think it was it Hooke who found cells for the first time? Yeah, in the 17th century. And for centuries, nobody knew what was really enclosing the cell like that. It was not until the 20th century that we got a good idea of what that was. All right, so long story short, we came to understand that phospholipids enclose the cell, right? And remember, phospholipids, like a lipid, they have a hydrophobic component, these two hydrocarbon tails, but they also have a hydrophilic component. You know, some lipids have that, right? And so they do have a substantial hydrophilic component. And so they have both hydrophobic and hydrophilic qualities, and their qualities and their shape cause them to form a lipid bilayer in water. So they spontaneously form a lipid bilayer, which is then the perfect structure to surround the cell. As we said, the cell being a packet of solutes, we need a lipid to contain them. And here's a lipid that won't just clump up, but it forms a barrier, unlike fat, right? So cool. What holds those two layers together again? Hydrophobic interactions. Very good, you got that one. And trust me, get it on the exam. Now, what really was the mystery? Once it was understood that phospholipids are the membrane, or main component of the membrane, what's the deal with the protein? Because we knew there's protein, but how the protein is arranged was a big thing. So I think it was really now into the 20th century, people were proposing this idea and that idea, and ultimately it wasn't until 1972, I was around at that point, 1972, that the correct description of the protein was provided. Okay? So what is the correct deal with the protein? The proteins are embedded in the lipid bilayer. They're embedded. It might be partially embedded, it might be completely where it's sticking out on both sides, but they're embedded in the membrane. Okay? Yeah, interesting. So, for example, this protein here has this component within this hydrophobic core, right? And then this part sticking out on either side. So, what do you think now about this protein? The parts that stick out would be hydrophilic or hydrophobic?
Philic.
Hydrophilic, right? So if you understand the blue is water, then yeah, that's in water. That's happily remaining in water, so it must be hydrophilic. That's exactly right. So what about these parts of the protein with the tails in the core of the bilayer? Hydrophobic, right. So these proteins, like the lipids, have both hydrophilic and hydrophobic characters, and those characters are sort of positioned where you would expect them to be, effectively stabilizing them within the membrane. Okay? Yeah, well, why don't the proteins just float away then? What's the deal? What's the best answer, maybe? Hydrophobic? Interactions, right. I mean, just like asking, why don't the lipids just come apart? No, they're held together by hydrophobic interactions. That's also anchoring these proteins within the lipid bilayer. Hydrophobic interactions. Okay, good. All right, so that's called the fluid mosaic model. And you look at it, and I hear some people, scoffing or something. I think it's unfortunate because, again, decades of work and consideration just to recognize that. Fluid mosaic, this was called. This was described as a fluid mosaic, and this is the fluid mosaic model, or fluid mosaic theory of the membrane. And like a good theory, it stood the test of time, right? So now for going on 50 plus years, we still believe that this is what membrane, we can't see it easily, but that's what it's got to be, because all of the data that has come since then has supported that model. Let's talk about these two aspects, because they're actually both important. Membrane is fluid, and by that it means movable, right? Movable and moving, like the consistency of salad oil, I think your textbook says. The consistency of salad oil, how fluid is that? So fluid, right? And so it's fluid, and you can see a rapid movement, actually, of the lipids side to side. Side to side, within each layer, side to side, or lateral movement, right? Lateral means side, right? No flip-flopping though. Side to side, but no flip-flopping. You don't get flip-flopping of lipids from one layer to another. They actually remain in each layer. Why is that? Why no flip-flopping? Yeah.
Then the deer will be exposed to the water, which is kind of different stuff.
Okay, so from a functional standpoint, I would argue with you that you're describing why there can't be any flip-flopping, right? Because if they do flip-flop, it would just mean that the structure is not very, resilient, right? It's not doing much for the cell if it's so dynamic in that way. And maybe, forgive me if I misunderstood, but I would say the reason it doesn't flip-flop is because it would violate the chemistry that we described, right? We said that this is hydrophobic, right? A hydrophobic core, but both sides of the membrane hydrophilic. Yes? Right, so moving a hydrophilic head through a hydrophobic core is not so compatible, right? Yeah, there's a very steep energy barrier, to put it that way. There's a very steep barrier for that to happen. It doesn't happen. So the lipids can move side to side, but not from layer to layer. Okay? Protein also moves from side to side. And this figure didn't show it, so I just added that. Proteins will also move, from side to side. Okay? So imagine now, the layer, the surface of the ocean, right? And then like a bob, right? Or a lure, kind of bobbing along. Like that's the protein, right? It can move side to side, but it's not going to move, it shouldn't move up or down, right? Okay. So what do you think? Is this fluidity important? We just spent 5 minutes talking about something that's not important, right? No, of course, yeah, it is. It is critical. The fluidity of membrane is essential for a couple of reasons in particular. One, the permeability of the membrane. Okay, we're going to talk about how nutrients get in and out. But if you're small enough, you can just go across a lipid bilayer. Okay, so some nutrients can just go across a lipid bilayer because the fluidity makes for a slight permeability. I'm not saying everybody does this, but some small substances importantly will be able to move across. Oxygen is a good example. If oxygen goes right in the cell, we don't have any particular means of moving them. Okay? It just goes right in. Number two, the proteins of the membrane must be able to move in order to do their job. Okay, they must be able to move. If you know anything about pharmacology, G proteins or I don't know, side-to-side movement is super important. We'll talk later about the electron transport chain, another example of proteins in movement side to side, and that's really necessary for them to do their job. Okay, so the proteins can function, you need that fluidity. All right, so this is really another example of homeostasis because you need fluidity. You don't want too much, or the membrane, is not doing its job. You don't want too little, and then you don't have these necessary functions, right? Okay, so how do you get just the right amount? Let's talk about this. Let's talk about what affects fluidity. Temperature, it turns out, affects the fluidity of the membrane. You can tell me, as the temperature goes up, membrane fluidity what?
Increases.
Increases, of course. Molecular motion increases, and so fluidity increases. Yeah, as the temperature goes down, fluidity decreases, right? That's exactly what happens. Okay, good. But another factor influencing fluidity is the type of fatty acid. The type of fatty acid. And what do I mean by that? I'm talking about in the phospholipids now. Excuse me. In the phospholipid tails, the type of fatty acid in the phospholipid tails. Okay, let's recall. Good review for the exam now. What are the types of fatty acids in phospholipids? So which of the two, left or right, which of the two bilayers contains phospholipids with some unsaturated tails? The left, right. So remember that an unsaturated tail in nature will typically have a double bond in a cis configuration, and a cis configuration double bond creates a kink in the tail. Okay, so we see a lot of kinks here, and that is because of the unsaturated fatty acid. And again, in cis configuration. By the way, that was a really challenging question, I could tell from the performance. So if you have questions about cis and trans double bond or how to tell, I direct you to my key for CDA number 3, right? But also address questions, please, before the exam rolls around, because you will see it again. All right, which is more fluid, left or right? Which membrane or lipid bilayer is more fluid, left or right? Okay, so what is fluidity, right? Is it a property of liquids or a property of solids? Property of liquids, right? And what is it about liquids? Are the molecules tightly packed or are they far apart? They're far apart, right? Connected with what you know, right? These lipids are pretty far apart, and we're not going to have so much hydrophobic interaction with these type of fatty acids, right? More fluidity. These type of fatty acids are really bunched together. Or the phospholipids are bunched together because of the fatty acid. You take my meaning. and that's going to be less fluid. More hydrophobic interactions and less fluid, okay? So which is more fluid? It's the one on the left, the one with the unsaturated fatty acids. Okay, this is a significant concern because if you can regulate your body temperature, then no problem, right? But if you cannot, are you just a victim? I mean, of the temperature, right? What do you do when the temperature goes up, as we know it does in Florida? What do you do, like, recently when the temperature
Are
goes
you a
down?
victim?
So let's say single cell life in particular, which cannot regulate temperature, can change the types of fatty acids they're using in their phospholipids. Okay, so as they build more membrane, you know, as cells grow and divide, they can alter the composition of their phospholipids, okay, or the fatty acid composition of their phospholipids. So give me an example here. So let's say the temperature went up. Well, a single cell life would probably make phospholipids with more what? Saturated or unsaturated fatty acid? Temperature goes up. Saturated. If temperature goes up, you want to bring down that fluidity, right? And more saturated fatty acid will be the way to do that. If temperature goes down, you probably want to make more what? Unsaturated. Again, bringing that fluidity back up. Okay, very good. Very nice. Okay, last but not least, folks, mosaic, right? Fluid mosaic. Why is it a mosaic, for heaven's sake? That's a nod to the protein. Okay, so membrane is made of lipid, but it's also made of protein, and so many different types of proteins. And remember, they are embedded in the lipid bilayer, although some are not. And I want to bring this distinction out. So those that are embedded in the lipid bilayer we call integral membrane protein. But some are only on one side of the bilayer, and those are called peripheral. Integral, embedded. Peripheral, stuck to one side. Last but not least, the proteins have a particular orientation in the membrane. What is that? Indulge me. It's a receptor. Okay, this is a protein that's a receptor. And you know, a hormone will bind here, and the receptor will convey that signal into the cell. Would the receptor function in this orientation? No. How does the hormone bind? No. So again, proteins have a specific orientation. It must be like that. Okay? So very important how the proteins are organized and their biochemistry. Fluid mosaic. Question? Great work, folks. I'll see you.